He previously was president of the Biophysical Society and a recipient of many awards, one of them being the Max Delbruck Prize for Biological Physics from APS. And he's very famous for his work on protein folding, specifically what's known as the HP lattice model. So this is a minimal statistical model of proteins. And more recently, in the context of non-equilibrium statistical physics, he's been working on what's called the principle of maximum caliber, which is the principle of maximizing path entropy as opposed to state entropy for non-equilibrium systems. So today, though, he's going to be talking, I guess, more about his work at the interface of biology and physics. So we're going to hear about what he has to say on the origins of life. Please take it away. Thank you.
Thanks, Sarah. It's really a pleasure to be here. This is a phenomenal physics department in my mind, and I'm really delighted to be able to talk to you today. I want to talk to you about the origins of life, and the picture here shows what we're looking at. On the right side, what you see is, let me see if I have on the right side is evolution. Everybody knows evolution. You came from your parents, they came from their parents, and so forth. And our species came from earlier species. You can roll the tape all the way back 4 billion years, and somehow we came from the very first cells. The singularity that we're interested in, however, is this arrow right here. Somehow, 4 billion years ago on the early Earth, it was all just simple chemistry, methane, ammonia, water, things like that, and spark discharges, something like that. And simple chemistry, simple physics, and so how did we get from here to the first cells? That's our question, origins of life question. Now, it's not known. It was a singularity. Nobody's ever reproduced it. There's no experiments that have been definitive, and so it falls to theory to try to lead the way here. But before I get into the theoretical models that we've been developing, I want to tell you a little story about a theorist and experimentalist. And I know this is going to ring a bell with a lot of you who are theorists. So it turns out there was this theorist and experimentalist, and they were condemned to die. They were in prison, and the judge comes to the theorist and he says, I will grant you each, he comes to both of them, and he says, I will grant you each one last wish before I execute you. And goes to the theorist first, and he says, and what is your last wish? And the theorist says, well, you see, Your Honor, I think I figured out origins of life, and I want to convene a community of all my colleagues and have a little seminar and symposium about, and I want to tell them the answer. And then the judge goes to the experimentalist and he says, and what's your last wish? He says, I want to be executed before that seminar. So this is one of these businesses where there's been lots and lots of speculations. This goes all the way back to Oparin and Haldane in the 1920s, and they were the first who more or less indicated, hey, probably life started on Earth, and hey, probably it started from chemistry and simple molecules. And ever since then, so that's 100 years ago, ever since then there have been all kinds of speculations and books and so forth. One very popular one at the moment is so-called the RNA world, the idea that RNA molecules somehow came first. There's this idea of the selfish gene. This is a very famous old book, 1976. The idea that DNA kind of somehow drives its own replication, the genes do. This idea called the blind watchmaker, the idea that this was taking issue with the idea that you could get order, the kind of order we have in biological systems, from disorder. And it would be like a blind watchmaker. How could you possibly imagine somebody assembling a watch from nothing? But one of the problems is that all of those speculations are fairly vague. They're not specific enough to tell us experiments to do, and they also are not a full story. They're like, if the RNA world idea is right, for example, it's one jigsaw puzzle piece. It's not telling us why did RNA start doing this, or why did there was other thing called metabolism first. Maybe small molecule chemicals came first. Why would they have then gone towards biology? Why would this have been such a persistent thing for so many billions of years? So on the one hand, there's the chicken and egg problem. What came first? What molecule came first? But the thing is, we need, in science, we need a bigger story. We need the whole thing. We need some kind of the entire puzzle, even if a lot of the pieces are wrong or out of place. We need some big picture into which we can now start fitting the pieces. So we need to know not just the who and the whodunit, but the how. What is the physical process? What is the driving force towards what? Why for 4 billion years has life been so stable? And that's what I want to try to address here, and that's a physics story, not an organic chemistry story. A lot of the origins of life work has been done by synthetic chemists who think about small molecules, think about how they might have come together on the early Earth under the conditions that there were at that time. There's lots of beautiful work that has been done by the organic chemists, but there's need for a physics at this problem too, and that's what I want to tell you about here. So our questions fall into the nature of the physics, the nature of the driving forces, and the nature of the dynamics. In particular, what was the force that pointed chemistry in some direction and kept it going? Fitness function. Biology has fitness function, so things get more fit. According to Darwinian evolution, what happens is you walk up fitness landscapes, and some species gets more fit than the other in the given environment. That's in biology. We don't have an equivalent of a fitness function in chemistry. Why would molecules start getting to be fit and be self-serving in some way? They lower energy, of course, but why would there be some self-servingness to it? And disorder. Now, one of the things that happens in the synthetic chemistry field is you do a clever experimentalist works on a lab bench and does an experiment, but the next day you walk into the lab, nothing remarkable has happened beyond whatever the PI was doing. And what we have to figure out is not how to have a clever PI tell us what molecule was interesting, but to have chemistry and physics itself do all of this. So this has to come from full disorder. And a key question is sequence to function. This is the key part of biology is the fact that it encodes all of our functions in protein molecules and RNA molecules and DNA molecules. These are all sequence dependent. And where did this sequence dependence come from, and how did that solve its sort of needle-in-a-haystack problem, if you will? So I mentioned life has two sequence polymers. This is really the crucial thing. We're going to focus on this. There are other kinds of mechanisms that people have proposed, like metabolism first, or maybe cell membranes first, and things like that. But as far as we're concerned from a physical point of view, you can't get to a living system without getting sequence-dependent polymers. That means either proteins or RNA molecules today. So this is how life splits up. Proteins are good at this part of life, creating functions, and RNA is good at this part, which is the information storage. Information storage is in the orange and the yellowish, and function is in the green. And the question is, how do we, both of these are necessary. Living systems you can't do without either the information or the function. But what we imagine, what we believe, the mechanism I'm going to propose to you, is that there is a way for proteins to get this started. It can get it started, but it will not get you all the way without RNA molecules too. So before we can develop a model, the first thing we have to ask is, what is life? If I need to be able to define the difference between biology and chemistry, I have to have some kind of a boundary here. And if I can't define the boundary, then I can't tell you whether my model is any good or not. So what is life? Well, so there's lots of definitions. It's hard to define life. There's lots of failures in trying to define life. For example, if you say that life is something that eats and metabolizes and duplicates, it turns out candle flames do that too. So that's not sufficient. So it turns out one of the definitions that is probably closest to a consensus, I would say, from the scientific community is this one from NASA, which says life is a self-sustaining chemical system capable of Darwinian evolution. Now, this is interesting and useful in two particular ways. First, it's quirky. because it's defining life not by, it's defining the thing, life, not by the thing itself, but by how the thing changes. Because the underline, that's our underline here, it says it's capable of Darwinian evolution. Now, most definitions of life do have something like that in there. It seems to be the case that you can't define life without talking about how it propagates itself. So this is quirky, but it's very important for how we think about the next step. So in short,
But that's,
yes.
so maybe don't worry really, but like imagine you took a bacteria
Yeah.
and you did something to it so that it was just any mutation would be fatal. It was incapable of evolving. It would still be alive.
Well, if it's incapable of evolving, then it dies sooner or later.
But you haven't changed the way it functions at all. You've changed what it can pass on to its progeny, but it itself.
So let me put this into a bigger picture, because on the one hand, life, as you define it in evolutionary terms, has to do with lineages, and it's not about the individuals, because individuals will die. And so we all have that same problem that you raised, for example. But the biology at the evolution level is not about us as individuals. It's about propagating our lineages and our species. And so we all have, and those die too. It's all a matter of time concept.
So in your definition, a living organism is not a reasonable concept, right? You can only have an organism that is a member of a set of living beings.
Exactly. So this is Darwinian evolution. So life is the process itself, and it's the populations, and it's the lineages, and it's not the individuals. We're all just sort of little players in the game. So in short, what we're looking for is an answer to this question. Life can't originate until it can propagate. And so what I mean by that, what we need for a model is to think about how molecule making in chemistry somehow became sustainable. And in short, what that amounts to is how the non-equilibria that were around in the early Earth and non-equilibria today couple to the business of molecule making. How did that happen originally, and how does that continue to happen? How do we connect up some pumping that's out there to making molecules? So the first point is it's non-equilibrium. Everybody knows this, but I just want to make a point, because this was raised in Schrodinger's famous book called What is Life back in the 1940s. The point he raised was there's a problem with entropy. He called it neg entropy. There's a problem with that, and that you can't get from chemistry to biology because you have to beat entropy. And what he did was really just raise the question. He didn't pose an answer so much, just in essence just raised the question. But clearly the second law was a big sticking point for him. And what I want to point out to you as we go along is the second law is not the relevant principle here, because the second law is about going to equilibrium. And it's certainly true that chemistry does, and it decays, and death wins, and entropy wins. But in biology, that's not the case, and I'm going to take a few more slides to say a little bit more about this. But the bottom line is, if you don't have a non-equilibrium pumping of some kind or another, you can't get life today, and you couldn't have gotten it back then. And so we need to be looking for what is this coupling? What is the non-equilibrium driver for molecule making? So let me first start with a little look at evolution itself. What does today's Darwinian evolution look like? And because this is what we're going to try to evolve from some microscopic mechanism ultimately. So we show this in the form of a kind of machine. This is a picture of Darwinian evolution. And we call it a machine because it runs in cycles and it's powered. So it's like being plugged in, is my metaphor, if you will. Here's, at time t, what I have is a population of cells, call them X. This is our wild type population. At the next time instant, what happens is there's a mutation in that population of wild type cells. And so we call this the momsample. This is a cross between a statistical mechanical ensemble and the fact that the ensemble is made up of mothers who make babies. And so the momsample is basically every possible mutation of the wild type. So from our ensemble, what happens is we get some mutant, let's say. And now what happens in the next time interval is that cell grows up into its own population. And now the two compete. This is the mutant and this is the original. And one of them wins. So this is the basic idea of Darwinian evolution. It's pretty well, it's standard, and I'm just putting this into terminology that I want to use for subsequent slides. But the gist of it is the winners then win more resources. And so this is a process in which makers make more makers, so moms make moms, but they're not identical. This is not a Xerox copying machine process. This is heritable variation is the term that people use in Darwinian evolution, which is to say you're not exactly your parents and they're not exactly their parents. There's variation, and it's the variation that's crucial. It turns out we can write some dynamics down that shows that Darwinian evolution would have died out if it was all about making identical copies. And the reason for that is because if you're an identical copy and you're perfect in today's environment, tomorrow the environment changes and now you're screwed and everybody dies out. So you have to have variation. And that's what this point is that you get change, and this change comes through mutations. There's competition among the makers. The winners, the alpha dogs, win. And a key point about this is if you're a winner, you sequester more resources to continue winning even more in the future. Your population gets bigger. So this is the nature of the machine. It's a growth and duplication machine. It's not a Xerox copy machine. So made a little bit more in the vernacular on this slide. This was a cool line I liked from a movie that nobody ever saw. It said, evolution forged the entirety of sentient life using just one tool, the mistake. So this is mutations. I always like that. And so the point is what we're looking for, when we're looking for how Darwinian evolution got started, how did this engine that's been running for 4 billion years, how did that engine get started? And what we're looking for is a machine that's like evolution. It's a disorder to order machine somehow. It's got to do mutations in some way or another. So it's more like a dumpster diver and less like a copy machine. Just to connect with those of you who think about population dynamics and so forth, it's pretty standard, going way back 100 years, to write down population dynamics. What population dynamics is all about is I have a population of moms that's the letter A is for mom, and the subscript N is for the different mom types and so on. And so what we're looking for is moms competing against moms and a resource base, and the moms utilizing the resource base. This is standard population dynamics, and it's a way representing competition and winners of resources. And so, but we have to modify this if we're thinking about origins, because for one thing, cells, once you have biology, you have cells and you have organisms and you have kind of recognizable utilizers of resources. Before that, you just have molecules floating around and you don't have any identifiable self. So we have to deal with the fact that our molecules aren't all collected together inside cells. We have to deal with that in terms of the concentrations here. We have to deal with coexistence. Not everything is winner take all. In Darwinian evolution, winner take all happens in the infinite time limit under certain limiting conditions. But in some conditions, what happens is you have a peaceful coexistence. And you have to have peaceful coexistence, meaning I need some backup populations. Maybe I'm the winner. Maybe I'm at the top of the hill. I've won all the resources. But if I don't have some other bacteria or other organisms that are at much lower populations who are still available, who are coexisting with me, if I don't have them, then they won't be able to pick up the slack once I get killed by a new environment. So we need this in there too. And the birth-death relations in most population models, many population models, are linear. That doesn't work either, because it would say the limit is if I go to infinity in the population, then I can get infinite birth rate. that's not true, and it turns out to be relevant for making this. So bottom line, you have to make some modifications, but largely these are just population dynamics models that we start from. But here's the most important point, and that has to do with how do I get out of chemistry and get into biology? In a generic way. I don't care what model you like, I don't care what molecules you prefer. This something along these lines has to be true. This is a lowest order expansion in dynamics. I've simplified now the previous slide into a growth term of moms of concentration A. There's a growth and there's a death rate, and the normal thing is, forget about the second term, the normal thing is I have birth-death dynamics. Now, in chemistry, D is greater than G, so everything dies. In biology, G is greater than D, so everything survives. As long as we have a birth rate that's better than our death rate, biology is always running. Those are the two limiting cases. One or the other of these two terms wins, and I normally have just birth-death kinetics. The thing that's missing, the reason I couldn't get from chemistry to biology in the Schrodinger world is because I'm missing a next order term that I have to have. And that next order term says, this is not just about moms making moms. This has got to be moms making better moms, making even better moms, making even better moms. And when you get to a certain point, you're making pretty good moms. Now you're up into the biology world where this term has now disappeared, and I am in the limit where birth is now greater than death. So I can get to biology, but to get to biology, I have to have this, we call it a cooperativity term, or a flywheel. We call it the flywheel. You get the flywheel started and then everything runs. So here's the phase diagram for this simple model. G1, growth over death, this is the normal thing, and what it would say is if I don't have a second term, it would just say, when D wins, I'm in chemistry, when G wins, I'm in biology. Very simple phase diagram. On the other hand, what the G2 term gives you is, it gives you a statistical barrier you can climb over. So if I have, even if the growth is small, as it will be in chemistry, as long as I have enough cooperativity, I can jump up into this green region, and from the green region I can get over to biology, and then I don't need it anymore and I can drop back down. I have to have cooperativity. That's a key thing, and it's true for any model you want to make microscopically. I need this second order term to be able to get out of chemistry and into biology. So now let's talk molecules and mechanisms. So the first point is that evolution works on, the normal phrase is, it works on phenotypes, not genotypes. What does that mean? It means it works on the protein end of it. It works on the functional end. It works on growth rate processes. It doesn't work on the information per se. The information, RNA and DNA, are useful for remembering stuff, but they are not the business end of what evolution cares about. And so the business end is functionality, and mostly that's proteins, and mostly that's because proteins can fold. So they have a single, unique, straightforward mapping from an amino acid sequence to a native structure to a specific function. RNAs don't, and I'll come back to that later. But that's important. You need native structures. This is the folding problem, from the amino acid sequence to the native structure, and then this is the function problem. What does it do for a living? But the bottom line is we need to be focused on protein functions if we're going to focus on what is it that evolution cares about. Now, point number one about in thinking about needles in haystacks is this is a problem that our statmech community already thought about before in the protein folding world, and this is what kind of got us thinking about origins in recent years. Was a problem that we already knew something about how to solve is this folding problem, and the origins problem I just want to show you is really resembles it closely enough that we could use a lot of the tools. Take a look at the top line first. What is the folding problem? Folding problem is this. I have an amino acid sequence that has, let's see what we got. N is the number of amino acids. Z is a number, let's say it's around 3 roughly. It's the number of conformations per peptide bond. But that's a ballpark number. And so the size of the haystack, if you want to think of needles in a haystack, The needle is the native structure. I'm looking for one answer here, one confirmation. From a haystack that's 3 to the n, where in this case n I took to be 130, which is lysozyme, small, simple protein. And so that's 10 to the 62 is the size of my haystack. 1 is the size of my needle. I'm looking for one native structure. This is a slight simplification, but ballpark, this is a needle in a haystack way of looking at protein folding, and this is how people did look at it back in the 80s and the 90s when we started working on this thing. Now, one way you can represent needles in haystacks in terms of energy functions is as a sort of a golf course landscape. And the golf course landscape means my degrees of freedom are left and right here are different conformations. And the needle, the one lowest energy state is the native structure. And if you think about this as the haystack having all conformations being equal energy with the exception that just the native is good, then you have this golf course shaped landscape in a very high dimensional space. Sorry, I don't think that was me. I think it was. Anyway, the golf course is a representation of the needle in the haystack problem. High dimensions, I'm showing only 3 here, but the bottom line is if you roll a golf ball along this, it'll take forever to find a hole. So that's how folding was looked at. This is also how origins has been looked at, except now the haystack, still proteins, still amino acids, but now it's amino acid sequences, not conformations. Now I'm in the sequence space, not the conformational space. The sequence is, at a given position in the protein, I have 20 possible amino acids. And now again, I have n amino acids in the chain, so now my haystack is 20 to the n, much bigger than this one. And now the needle that I'm looking for, I have many needles in this case because I'm looking for different protein sequences that fold up to useful biology. But again, it's very much monkeys dancing on typewriters to type out Shakespearean plays and things like that. It's all the same metaphor. Oh, I see what I'm doing. Sorry about that. Oops. Okay. Anyway, this metaphor is wrong, is the problem. Needles and haystacks aren't relevant here. Statistical physics is relevant here, and statistical physics always can solve these problems because when you have energies and you have cooperativities and so forth, it tells you how you get order from disorder, and that's what I want to describe here. First on folding, what did we learn and how do we parlay that into thinking about origins? We learned a couple of things. First of all, that the haystack is not 20 to the n. It's essentially 2 to the n. It's a binary code. It's largely hydrophobics and polars that dictate what the fold structure is going to be of a protein. I don't need to know which of the 20 amino acids. I just need to know is it red or blue. And roughly speaking, it's about 50-50. In today's proteins, it's about 50-50 hydrophobic and polar. Now that was the model that we made these little lattice models back in the early 90s, and experimentalists then did the experiment to test whether we really would get a reduction of 10 to the nth, so 130 orders of magnitude or something, by that simple recognition that the physics is really just binary and not a 20-letter code. And so this was Michael Hecht's group, and also Bob Sauer published papers that show that if you mutagenize 4 helix bundle proteins and randomize the interior and the exterior, keeping only the binary code, making sure that you have polar on the outside and hydrophobic on the inside, that you do get pretty much the same structures. And the other thing that came from a lot of this lattice modeling was these are not golf course landscapes, they're funnel-shaped landscapes. Bumpy funnels for sure, but still they're roughly funnel-shaped. What does that mean? It means that the top of the funnel is big conformational space. It's wide, so that there's a lot of entropy, big conformational space. If you make any contact at all, you go down the funnel, which means I have better energy. Down is better energy. I have better energy, but I also have fewer conformations because we've already nailed something together. And then make another contact, I've nailed something more together, and more, and I reduce the entropy. And so these things are funnel shaped, and it tells us in essence that almost anything you do, as long as you keep making contact, will take you closer and closer to a very small space to search. All right, so now for origins, the challenges are the following. First of all, what the organic chemists have told us is that you not only can make amino acids on the early Earth, this is the Miller-Urey experiment of 1953, and now thousands of other experiments that are related to it. The gist is take small molecules, a lightning strike, for example, and you can make amino acids. And not only make amino acids, you can make nucleotides too, and you can make a number of very simple biological molecules. That we know, we assume that's true. We also assume what's also known is you can string them together, make covalent bonds. Even meteorites these days have peptides of order 6 or 7 amino acids long. They're not that hard to make. Peptides are much easier to make than nucleotide chains, but still, bottom line is, this is not a difficult thing to get. What is a difficult thing to get is long chains, and that's always been the bottleneck in the synthetic chemistry world. Shown in the cartoon here, I have some monomers. I have some, this is a catalyst of some kind. Let's call it the founding rock, some big rock in some lake in Nebraska somewhere. This is whatever is the catalyst that is running this, helps make peptide bonds, and so you get short chains. But what you see on the right is when you do that in the organic chemistry experiments, the chain lengths are shown here on the x-axis. The y-axis is the log of populations. You always get this, you could call it the Florey problem. Florey discovered a long time ago that when you polymerize chains, invariably you get many, many short things and very few long things. And that's what this is showing here. These are for amino acids and nucleotides in these experiments. Invariably, the challenge is getting to long enough chains, and if you don't have long chains, you don't get proteins, and you don't get folding, and you don't get catalysis. Now, one of the problems, however, is that the polymer world has normally thought about 2 classes of polymers. One is those that are all polar or charged monomer units, and so they dissolve in water, or others that are all non-polar. They don't dissolve in water, and they make things like carpets, for example. So there's 2 different classes of polymers that the polymer guys have looked at quite a lot. But one class that they did not look at are these copolymers of specific sequences of hydrophobic and polar residues. Proteins have them. But when you look at these, first I'm going to tell you about theoretical models, computational models that we do. Some of them are pretty useless. They don't do anything. They don't fold. The all polar chains are just floppy, and the all non-polar chains just aggregate and make tar. That's not useful. All right. But some of them fold, and the reason is very simple. If I have 50-50 oil monomers and water monomers, the thing is, the oil isn't going to like water, and so it's going to collapse. So you get a lot of collapsed chains. So random sequences, in principle, in the computer at least, I'll show you experiments later, in the computer what happens is random sequences of 50-50 hydrophobic and polar just have to fold, have to collapse because they need to get the non-polars out of water. So there will be a lot of these kinds of things. And by the way, that scales with chain length. That's still true if you get even long chains, because 50-50 still means I got a lot of hydrophobes. And some of them that fold also will catalyze. They'll have surfaces. The surfaces will be relatively stable. They'll have amino acids on them, which are all kinds of chemical moieties, and they can catalyze reaction. So our picture is very simple. It looks like this. Early Earth, what happens? I have hydrophobic and polar monomers. I'm in some rock in Nebraska is my catalyst. I make these little short peptides. Some of them fold up because they have the hydrophobes in them, and some of those become catalysts as well. Short peptides can catalyze some reactions primitively. Now these things will be, we call them yarn balls because they're not really specifically folded to native like structures. They're kind of more randomy. And these will elongate, however, and the reason is there are two forms of cooperativity. Remember, I used this term G2 before. I said we need a cooperativity. And a molecular model, you need the cooperativity. Where is it coming from? The bottom line in this model is it's coming from two things. One is, once a chain folds, it dies slower. Why? Because it's compact. So water and all the bad stuff that would get in and break down a peptide chain can't get to the inside. That's well known. Anyway, it's the physics. It's one of the two cooperativity terms in this model. It's very simple physics. And the second cooperativity aspect is the fact that some of these will have some catalytic activity. They have chemical moieties, they're compact. I'm going to show you a little bit more about that later. But the gist is, if it catalyzes, then what happens is this whole process gets driven to the right. And what you get in the computer, and this is a computer model up here, is this is that Flory plot again, but the green and the gray are showing you normal polymers, and the orange is showing you polymers that have these HP random sequences, and therefore the compactness, and therefore some level of catalytic capability. What you can see is this extends out and gives us longer chains. It also gives us an autocatalytic-like process, not perfect Xerox copying, but autocatalytic sets. Anything that folds up and has a catalytic surface on it can help elongate chains that themselves are also catalysts, and so you get these autocatalytic sets. So this has some ingredients that can solve some of our early problems. It elongates chains. It starts giving you sequence effects for defining folds and functions, gives you autocatalytic sets, and it keeps going. Now, the problem is we're still way over on the disorder side of all of it, but we're now moving towards the direction of enough cooperativity that can get us towards that tipping point. And you get order from disorder in the sense that what this is it's really just a garbage generator. So you make random peptides on this rock in Nebraska, and mostly what you get, this is law of the population here, mostly what you get are very short random chains that do nothing at all. Totally worthless. That's the overwhelming population that you get. But a small fraction of those are longer chains and more collapsed, and longer chains and more collapsed and more catalytic and so forth. So you get a funnel, now not in conformational space, this is a funnel in sequence space. So, can we test it experimentally? There are the three main questions here that we're able to take a look at in principle are, do random HP chains collapse? Do they catalyze peptide bond formation? And do you gain chain length and stability from them? And again, this is the toy model version of that, but now what we want to do is experiments on this. The experiments we do are in these molecules called peptoids. That's not a typo. Peptoids are not the same as peptides. This is a kind of molecule. Here's what a peptide looks like. Here's what a peptoid looks like. The peptide has the side chain, one of the 20 different amino acids, on the C-alpha carbon of a peptide backbone. And what you do if you wanted conceptually to turn this into a peptoid is you pluck the side chain off the C-alpha carbon and you plug it onto the amide nitrogen instead. And so now your C-alpha carbon is no longer chiral. So you don't have chiral chains anymore, but you have the same amino acid sets of potential side chains. So you have a very protein-like molecule, same side chains, same hydrophobes, same nonpolar and polar residues, but you have a different backbone, and so this is the guy, Ron Zuckerman is the guy who invented the synthesis of this, published it in 1992. He worked at that time in a company called Chiron. Chiron made these polymers because they were interested in making drugs, and they thought, here was the secret, they thought that was magic, is that if you could make peptides now, if you could make peptide drugs, they would be spectacular because you could make them quite specifically, but you put them in the body and they get degraded immediately because the body recognizes them and breaks them down. So peptides in principle would be perfect drugs. In practice, it's very hard to turn them into actual drugs. And what Chiron thought they were going to get here was something that looked like a mimic. It was a sleuth. It was something that would fly below the radar screen of the hydrolysis because it doesn't have the peptide backbone, so that's not going to get chewed up anymore, and yet I can make side chains and sequences. And they thought, gee, this is great. We got a whole new class of drug candidates. Bottom line was Chiron decided that wasn't going to work. They got out of the business. They were done. Ron was working there as a bench scientist. Fortunately, he figured out how to fly below their radar screen. And so he was able to continue making these peptoids. My wife worked there at the time. She introduced me to him, and Ron and I decided, hey, instead of making small molecules and trying to make drugs, why don't you make long molecules and try to make like protein-like things instead? Wouldn't it be cool if you could make foldable polymers that were like proteins but didn't degrade? So Ron and I started that back in the 90s. And the great thing about peptoids, which I'll show you on the next slide, the great thing about them is they're polymer versions of proteins. They don't have the specific, you don't have a lysozyme specifically doing lysozyme things, but I can make sequences. I can make H&P sequences, any sequence I want. The polymer community at that time couldn't make specific sequences of anything. They could only make block copolymers or random copolymers or homopolymers. They couldn't make sequences, and Ron's big breakthrough was he could make these things into specific sequences. So you can control the sequence, the chain length, the hydrophobe type, the composition, all that kind of stuff in these molecules. And so they turned out to be great models of what random proteins would look like. And they're perfect for exactly what we need here. So this was what we've done over the last 19 years. That's Ron again. This is your buddy here, Kent Kirschenbaum, who's in the chemistry department here. He's not here today, but he was a grad student in my lab at the time, and now he's kind of Mr. Peptoid. And Annalise Barron, who's at Stanford, and she also is working on these things. The peptoid community is now pretty big, and they work on all kinds of materials, biological and otherwise. But here's what wouldn't matter to us for the present purposes, is that in these various papers over time, what was discovered is that these things are very protein-like. You can, as I said, the synthesis was 92. You don't need the hydrogen bonds in a backbone of a protein to make a protein-like fold, it turns out. The H&P sequence is sufficient. Because the thing is, once you move the side chain over to the amide nitrogen, you've screwed up the donor. You don't have a hydrogen bond donor. And that was great for us because we wanted to know how much protein folding is about H&P versus hydrogen bonds. Well, he could do it here because he didn't have hydrogen bonds. You can't disappear them in a protein. You can disappear them here. You don't need backbone chirality. You can get the chirality back through the side chains. You can just dope a few side chains with chirality, and you can get them to be chiral, but intrinsically they're not. You can make helix bundles, you can make zinc fingers, you can make things that are very protein-like. So we found these peptoids to be super useful for thinking about protein principles, and that's how they're useful here. So here's what we do. The picture on the right is showing you just a reference problem. This is to show you what a Kratky plot looks like if you take a protein, an actual protein, and when it's denatured, you get a signal in a Kratky plot that looks like small angle X-ray scattering. You get a signal that has sort of a plateau here and then goes up at the end. When you have a native protein, what happens is you get this big dip instead. And you can even see this now in time resolved work. You can take a protein and watch it fold, and you see all this stuff develop. So this is my reference state for showing you what happens when you take these yarn balls. And by yarn ball, what I mean is now I'm taking a 50-50 composition of H&P. I'm fixing the chain length. In this case, I think it's 50. It's 50, yeah. Fixing the chain length, and I'm saying, what is the entire ensemble of every sequence, the whole sequence space? That's what I'm calling a yarn ball, is the whole sequence space. I'm asking, does that sequence space of these H&P random sequences look anything like an actual single specific protein? And the answer is, at many levels it does. for example, this is the Krotkey plot that shows you if you take the all polar sequence, which of course is just open and floppy and doesn't do anything interesting, looks like an unfolded state, you get something that looks exactly like an unfolded state. On the other hand, if you take the 50-50 hydrophobic and polar, what you get is something that looks like a folded state. So this is the whole ensemble that's essentially collapsed.
What are the units that you have on the axis?
Oh, sorry. Yeah, the scattering angle and RG is the radius of gyration. Radius of gyration.
Oh,
This is the form of a Krotkey plot. Basically, it's what I'm illustrating over here is just when you take this complicated square quantity of intensity versus the scattering angle, basically what happens is it distinguishes for you what an unfolded state looks like versus a compact state. And I'm just showing you that in this case, the whole ensemble of these yarn balls looks exactly like a protein does. And also, it turns out in denaturant, I have zero, in this case, we use acetonitrile as a denaturant. Low denaturant, you get something that looks native-like. High denaturant, what you get is something that looks unfolded. This is the whole space. This is the whole sequence space for that composition and chain length. So what this is saying is, we're not looking for like some little white dot in a black sea. We're not looking for a single sequence like lysozyme would be when we're looking through sequence space. We're getting protein-like properties here from practically everything having that composition and length. Question 2 is, does it catalyze reactions? Well, so far we don't have an actual peptide bond formation reaction, but we do have a bond formation reaction of this particular chemistry. And what I'm showing here is the rate. This is time, so you're seeing the slope of this is the rate of the uncatalyzed reaction. This is just the normal reaction in water. And here is the reaction when I put yarn balls in there. So I'm asking, are the yarn balls actually giving us something like a catalytic process that helps accelerate the bond formation? And you can see that it does. And so the question then is, so how are these yarn balls actually acting in some way as catalysts? And I guess our thinking at the moment is it's something like this. This is a kind of object that when you put into water, turns into an ensemble of objects. And the ensemble of objects is different cavities of different sizes and radii and different hydrophobicities and different dielectric constants. And so inside all of these little yarn balls of different sizes and shapes, two molecules A and B can go float into them, and the ones that happen to find a good dielectric medium for what they want to do, they react. And so this is basically a sort of an ensemble of solvents. That's what it's giving us. It's an ensemble of solvents. That's what these silly little things are doing. They're not native proteins doing native protein things. They're yarn ball things doing yarn ball things. But those yarn ball things are catalyzing reactions. So summarizing this bit before I wind up, our picture is this, that we have this catalytic rock in this lake in Nebraska, monomers making short chains. The short chains are able to catalyze and make longer chains for two cooperativity reasons. One is it buries a core, so it degrades slower. And second is it can catalyze reactions. Now this thing continues going on, according to our mechanism, to the point where there's an untethering event. This is a transformative thing that would have happened or it must have happened in origins, whether by this mechanism or something. And that is the following. I'm starting with a rock in Nebraska. I have this macroscopic rock. It's going to do a single kind of catalysis in a single kind of way. And what I end up getting is a huge diversity of catalysts and action mechanisms from these proteins. And when it gets to the point that these things happen to be good enough for catalyzing themselves, I have now untethered catalysis from Nebraska and made them available in miniature form, tailorable for any reactions anywhere. And that's a very transformative event. Now, by our mechanism or by some other, untethering was a very important step, I think, in origins. And this is just a reminder that we do have some evidence that these steps happen. We don't have evidence yet for the elongation bit, for example. I haven't produced a peptide bond from any of this yet. Just to end up with answering a few questions that often come up. Why not RNAs instead? Some RNAs are known to be functional. This is a list of them, for example. tRNAs are important. Ribosome, the RNAs in ribosomes are functional, and so on. There's a bunch of ribosomes that do have function, and that's because they have structure. Proteins have sequence structure function. Mostly RNAs have sequence information. That's what they do. They don't have structure, and they mostly don't have function. Lots and lots of RNAs these days are being found that do have function, but almost invariably they're partnered up with proteins. So yes, there are some. Perhaps RNAs could have been a start for this, but again, this is the summary of reasons why proteins are better at doing protein stuff and RNAs are better at doing RNA stuff. For one thing, proteins are not only compact, but they also make a little flower garden of different chemical moieties. You've got 20 amino acids. They can all sit there and perform catalysis in a complex way. What happens in RNAs is you bury all your useful stuff. You make these train tracks. You have hydrogen bonds and base stacks. That's all you get. No chemistries, and it's buried, so it doesn't face the world. It can't do anything. Now I'm oversimplifying, but to first approximation, that is why proteins are functional molecules in biology today, and RNAs are not. And let's see, okay, and yeah, and this point also is interesting. If you look at the protein data bank, which also has RNA structures in it, it has structures in it, biomolecules in it, but only 0.5% of the PDB structures are RNAs. It's just that most RNAs have very bumpy landscapes. They have lots of deep wells and lots of high barriers. They're very complicated. They don't have single structures, so they don't have single functions specified by a sequence. And there's good physical reason why biology chose the division of labor it did, and that is because proteins are terrible at information and RNA is terrible at function. And so they serve their purpose. Why are proteins terrible at information? Because information, you need to stretch it out to read it. And proteins don't stretch out. That's not what they do. They fold up and they ball up. You can't read it. So proteins would be terrible at the information function. RNAs are terrible at the functionality function for reasons I just mentioned. Bumpy landscape, lots of conformations, they don't lock in. And when they do lock in to a single conformation, RNAs, they're often partnered with proteins, like in ribosomes, for example. The rest of the story is, I told you the fold cat story, the yarn ball story, if you will. This is only a part of the story. This only sets the table. This is not, I wouldn't even call this fold cat story an origins life story. I would call it a pre-origin story, because there's a whole lot of stuff it doesn't do yet. I don't have RNA in here. I don't have any information in here. I just have proteins. So I don't have cells and membranes. I don't have metabolic networks. I don't have an encoding of protein synthesis, no genetic code.
You don't have self-replication yet.
Sorry, say?
You don't have self-replication.
Right, I don't have, I'll show you replication, to your point, I'll show you that in a second. But interestingly, this does satisfy NASA's definition. Now, which says that life is a self-sustaining chemical system capable of Darwinian-like evolution. Okay, so you could say, well, okay, does that mean it's alive? And I say, no, that means NASA's definition needs some work. They got it wrong, not us, I think. And so I think you need more. But let me come to your point right now, and that is, here's the thing. In biology, we have these beautiful streams that go from grandma to mom to kid, and another string that goes, all these lineages. Every lineage has its own identity, has its own DNA identity, and lineages of families or lineages of species or whatever. Oil droplets are the opposite extreme. This is sort of order to order, if you will. You know, grandma has some grandchild. This is disorder to disorder. Oil droplets can split, but there's no traceability. You know, an oil drop that came from some parent oil drop, I got no way of knowing what parent that came from. There's just oil drops are just oil drops. They all split. There's no address. And this is sort of in the middle. Fold cats have these have this constricting ensemble. So they'll argue you get lots of junk at the beginning, less junk, less junk, less junk, and so on. Now that's at the fold cat stage. This would be at the fold cat stage with just protein. If I need to get from here, from the fold cat stage, now up to biology, where I have really closer to a Xerox copy machine, now I gotta have RNA and DNA. I don't have that yet. And what that would do is it will constrict this in some interesting, complicated way. I have no idea what that looks like. My last 3 slides are just to sort of summarize, because there's been a lot of things I've tossed around here, and I want to just sort of summarize them in 3 parts. First of all, evolution. What is evolution? It's the non-stop engine that's been running for 4 billion years. It's like a Monte Carlo thing, but it's got a unique search space. Now, lots and lots of things are Monte Carlo-like. So most of statistical physics is about thinking about Monte Carlo-like things in molecules and so forth. And a lot of reinforcement learning and deep learning is all Monte Carlo-like stuff. And all kinds of probabilistic optimizations are all Monte Carlo-like. So that's not special. Evolution is not special in the sense that it's doing a random sampling of something and making a selection based on fitness. That's a kind of a very normal sort of thing to do. What's unique here is the search space. The search space, the search mechanism, and the fitness. Now, and so it optimizes. It steps randomly and selects on fitness. The search space is interesting. It's not molecular energies, as you would get in statistical physics, not molecular momenta or positions or conformations or something. That's not the search space here. The search space here is also not a machine learning thing of looking for patterns of some kind or another. The search space here is catalysts. The search space is also not small molecules. Evolution doesn't work directly on small molecules. What it works directly on is catalysts of small molecules, and that's super powerful, because that means you can rewire, what biology can do and did do, is it can rewire chemistry at will. It can make whatever chemistry you want happen by converting one small molecule to another. It doesn't do all of them, can't catalyze all of chemistry, but it does a lot. And so this is a very powerful search space, because you're able to create material. The fitness is differential growth functions and phenotypes, as I mentioned, so proteins mostly today. The search process is ergodic. What I mean by that in this context is that any mutation you want to make has to be accessible to be made, and without much bias relative to any other mutation you would want to make. You want to be able to search a whole space, whether a mutation is good or bad. You don't want to care about it when you're making the search. You want to care about it only when you're making a fitness decision. And the search process here is changing sequences, and sequences that I can store in RNA. And RNA and DNA are perfect for storing information because they don't fold, they don't have sequence dependence, whereas proteins have a very strong sequence dependence. So that division of labor is very important. And so, as I mentioned, its power, the power of evolution is it's able to create and rewire as much of chemistry as it can handle. That's a very powerful thing. Second is what got it started. What was the flywheel? So our argument is, first of all, biology today is that birth is greater than the death rate. And as long as you've got that, you've got continuing propagation. But origins didn't have that. Origins, birth minus death, was less than 0. If it's chemistry, it dies out. But with the cooperativity term, you can make it greater than 0. So I can get from chemistry here to biology through a process that involves some form of cooperativity. Moms have to make better moms and better moms, and so on. And our 2 cooperativities in this modeling are folding and catalysis. Now, the folding and catalysis, one of the arguments these days, well, actually, let me come back to this. Folding and catalysis are the mechanisms in the model. The first fitness was persistence. What was the fitness back in chemistry days before biology got a hold of it? It was just persistence. It's just life in the environment. How long do you live? How long do you survive? And in this case, folding stability is sequence dependent, persistence. So it's the first fitness function. After after a while, persistence isn't the only thing that becomes important. Once you get to biology, there's all kinds of ways you can be self-serving. But at the beginning, it's just how long can I live? And the answer is the better sequences are the ones that fold better and catalyze better. And the goal, what was the goal? What was it trying to get to? So it's not that chemistry was trying to get to biology. It's that chemistry was trying to get to persistence. And it turns out biological molecules configured the way they are in cells happens to be a phenomenal way of getting to persistence. Last point is physics principles. Are new ones needed? So there's a view out there at the moment in the world that this problem requires new physical principles of some kind. Our view is that no new physical principles are required at all. HP polymers fold and catalyze just like proteins do today. So proteins just did then what they do today. I'm not invoking anything new. They fold, they catalyze, same forces, it's the same physics, it's just oil-water physics embodied inside protein chains. And then the question of are any physical principles violated? No, the second law of thermodynamics is not violated. It's not totally irrelevant. It matters in chemistry, but the real point here isn't what's happening when you're in chemistry. It's the cooperativity you need to get out of chemistry, and that's not really a second law issue at all. Disorder to order. Again, this is really a statistical physics thing. As long as you have cooperativity and you've got some kind of a fitness driven by non-equilibrium, there's not really a problem. I plug in my television set, I get a very ordered state of things for a long time as long as it's plugged in, and that really has nothing to do with the second law per se. The haystacks are not so big. Yarn ball space itself has lots of needles in it, sort of primitive versions of them. And the tipping point would be or at least one important tipping point would be when the catalyst became untethered from the macro scale one that had rigid functionality and now has controllable functionality. Last point is these are the people that did all the work. And I want to particularly point out Ron Zuckerman, who was the developer of peptoids, Don Wei, Chang, and Sam also were doing the experiments I mentioned. And Charles Coker and Swalem are doing the theoretical modeling these days, and thank you for listening.
Are there any questions? No. Okay. Mark, yeah.
Jan, thank you. That's really, really beautiful. So I was sort of waiting for a little bit of a punchline, I guess, that would kind of help us understand how do we get to like that next step in the central dogma of biology. How do we get to RNA sequences that code for the right, amino acid sequences, and then. And that's a long sequence space. So we know there are a lot of RNAs that are non-coding; they do nothing. But biology has found some use for them because they're there.
Yes.
So how do we get to that? I guess this is sort of a next step for you. I don't want to, take us too far off topic, but I'm sure you thought about it.
We've thought about it. We don't have much, but this
Okay.
is what we have so far.
Yeah.
It is absolutely the next question,
Yeah.
and still puzzling over a lot of it. But here's what we have so far. The basic idea is why. If this protein world is so cool, why do RNAs come in? What's the fitness function? What's the cooperativity? What's the flywheel? Why does RNA and protein end up being better than just protein world? First of all, the protein world is actually pretty crappy at the level of its own death is sort of on a molecular time scale. Even with this whole FOLCAT thing that I've got going, you're dealing with molecules, and these are molecules that will break down on the time scale. It's molecular. They'll hydrolyze. So the power of having things like cells and heritability are that you extend the time scale, the persistence, if you will, the fitness, from the chemical time scale of degradation to almost infinity. Because a species or a lineage just goes on and on and on and on, and it just can carry on the process forever. So persistence can be huge. Now the question is, so how would it happen? This, I think, is sort of the simplest way of looking at the RNA protein, today's RNA protein world, and that is that nucleic acids make nucleic acids, and they use nucleic acids as catalysts. Some of them do. And the RNA molecules are needed in the ribosome to convert amino acids to protein, and then the proteins are needed here. So you see this as the sort of at your hand drawing a picture of the hand. It's this feedback. It's complicated, but if you can, so the thing that we need to get to for thinking about the genetic code in the RNA protein world is how do these things come together and start talking to each other? We have a model, and the model is basically putting this into dynamics. And I'll tell you the one result that the model gives, but first let me just tell you what it is. What it says is the following. Proteins do just what I said they do before in this fold cap model. They function. They have a fitness. That fitness is persistence. How long do I live? That's what proteins are doing here too. Same model, same parameters. What is the RNA doing? The RNA is not doing fitness things. In fact, it's doing sort of the opposite of fitness, but what it is doing is it's doing the Xerox copy machine thing. So imagine the following. I have a biocondensate. That's RNAs and proteins. They're all coming together, and there's a unit of them. Now, protein has functional fitness. The RNA is making sure that this object has a daughter. And if I get, so it's the fidelity that the RNA is giving me into the next generation, whereas it's the fitness that the protein is giving me. And it turns out the one result from this dynamical model of this is that this is showing you a sort of 1/x function. This is the fitness on this axis of the protein. This is the fidelity of the RNA on the other axis. It turns out it can double bootstrap. If I have a crappy RNA molecule, I can make up for, if I have a fairly good, let's say I have a fairly good protein molecule and a crappy RNA molecule, it can make up for it. It can bootstrap a bit and vice versa. So what I don't need to do, Manfred Eigen years ago wrote of this error catastrophe. The error catastrophe is about a single type of molecule, either RNA or proteins or whatever. And what he was saying was really the bottom line is they're all going to die. And so how do I get over this death threshold? Well, the answer here is you have a double bootstrap. My proteins may be crappy, and then my RNA can pick up for it, or vice versa. And there is an extent to which you can make that trade-off. This is a Pareto formula. There's a lot of stuff that still needs to happen here, and I don't have mechanism and so forth. But I think the bottom line is RNAs give you a huge advantage compared to this protein function world because they give you an address into the future. They give you this heritability, this lineage bit, and that's super important. That's about fidelity, though. The proteins are about function. I think the protein function you have to have before you can get evolution going. Then you need the RNA to make sure you can actually propagate.
So in your view of the origin of life, it started with the proteins then, as opposed to the RNA?
Yeah.
And one thing that, I always wondered about, for example, the ribosome, there are very few enzymes which are made up of both RNA and protein.
Yeah.
And it seems like you need at least, some of those in order to talk between these two stages of biological matter, right? So do you have any intuition on why it is that we only have, we have so few enzymes like that are ribozymes?
What I think is that the value of the RNA in those cases is in interacting with other nucleic acids. So RNAs often are interacting with DNA or other RNA molecules, and that's great because there's complementarity and hydrogen bonding and base stacking, and that's very important. So you need RNAs for some of those things. But in cases where there's an RNA version of a protein enzyme, the protein enzymes are sometimes thousands-fold more effective as catalysts. But still, for certain functions, like holding on to RNAs and so on, you need an RNA molecule. Now, the reason that they invariably come with the protein attached, I think, is that RNAs are negatively charged. I need a positive charge to be able to just charge neutralize, so I take positively charged protein. In the ribosome, that's what you find, is They're all little plus charged. The surroundings of the mRNAs are all little plus charged things. And I think that the thing that proteins do for RNA is to make them be catalytic. The reason they need to work together is because RNAs have so many minima in their energy landscapes, they can't really decide what they want to be. You know, do I want to be this fold or this fold or this fold or whatever? And what proteins can do is they can say, we're going to lock you into this particular fold. And then once the proteins lock the RNAs in, then the RNA can be the business end of a catalytic reaction.
Okay, unless there are other questions, there are refreshments downstairs in the CSMR area, so I welcome you all to come and join and ask more questions. Thank you.